You might have heard regarding the MACO. The MACO is maximum allowable carryover in the cleaning validation. This term is very important and careful calculation is required for the cleaning validation. MACO is maximum allowable carryover. Welcome to the video, and in this video we are going to learn regarding the MACO calculation in the cleaning validation. Please know that in the pharmaceutical industries, many of the products are manufactured. The facilities are used to manufacture many formulations, many drug substances. So it is not like that in the one equipment or in one area or manufacturing area, only one drug product will be manufactured. So the manufacturing area is used to manufacture many formulations, many drug substances. So there is a chance that if there is improper cleaning or improper cleaning validation or improper cleaning procedure developed may lead to cross contamination. That means if you have manufactured a batch of, let's say, formulation A, then cleaning is performed and then the manufacturing of product B is started. So there are chances that the residues from the manufacturing of product A may contaminate the product B. This is the simple understanding behind the requirement of proper cleaning and doing the cleaning validation. So whether the cleaning validation process developed is okay, suitable or not, and whether the cleaning validation is complete or not, that becomes very important. Cleaning validation ensures or establish the documented evidence that the cleaning process, the cleaning activities are performed as per the requirement, and there is very less chance or there is no chance of the product contamination because of the manufacturing of many formulations or multiple products in the same area. And then the cleaning validation involves the term MACO. This is maximum allowable carryover. So this is the safe amount of the drug substance mainly which may get into the another product and which will be safe. It will not lead to any toxicity. It will not decrease the safety of the another product, and it is calculated based on the scientific considerations. If you have any other thought or if you have any query, you can comment. Now start with the MACO calculation and understanding about the MACO. Cleaning validation involves the term maximum allowable carryover, that is MACO. It is required to be calculated, and MACO calculation is based on three terminologies or three scientific rationales like therapeutic rationale, toxicological rationale, and the 10 ppm criteria. MACO, it is a maximum allowable carryover, and MACO is the quantity you can consider which defines the maximum amount of residue, and residue is of API from the previous product that can be safely carried over into the next product without risk. For example, if you are manufacturing a paracetamol tablet batch and after paracetamol tablet batch you are going to manufacture metformin tablet batch, so MACO will be applicable for the paracetamol, the quantity of which getting carried to the metformin batch. Then the MACO quantity should be such that, or it should be limited, or it should be controlled in such a way that if it is going into the next product, it will not produce any risk. and that's why MACO calculation comes into the picture. Basis for the MACO: therapeutic dose, toxicological data, NOEL and LD50, and 10 ppm criteria. Many of the professionals consider the 10 ppm criteria, and out of these three bases, lowest value should be considered. Also, many of the professionals do the MACO based on the worst case scenario as per the product manufacturing facility. So first is MACO based on the therapeutic dose. In this calculation, the therapeutic dose of the previous product is considered, that is worst case. Smallest batch size of the next product is considered. Therapeutic dose of the next product is considered, and safety factor, typically 1000, is considered for the oral formulations. MACO is TD of previous into BS divided by TD next into SF. SF is the safety factor. Second is MACO based on the toxicological data used when the therapeutic dose is not available. So in this, two steps are there. Step 1, calculate NOEL, that is no observed effect level. Step 2 is calculate the MACO. NOEL is LD50 into 70 kg body weight of normal human adult divided by 2000. Then MACO NOEL into MBS divided by TDD into safety factor. LD50 is lethal dose in gram per kg. MBS is minimum batch size of next product. TDD is largest daily dose of next product. SF is the safety factor. So this is the calculation based on therapeutic dose and based on the toxicological data. Now coming to the 10 ppm criteria for the MACO calculation. MACO is equal to 10 ppm into smallest batch size of the next product, and batch size is considered in the kg. This is a general industry default when no therapeutic or toxicological data is available. So compare with the other methods and choose the lowest MACO value. Comparison of MACO approaches. So approaches are therapeutic dose approach, toxicological approach considering the NOEL and the 10 ppm criteria. Therapeutic dose basis is it is based on the patient safety and used when therapeutic dose are known. Toxicological criteria is considered when therapeutic dose is unknown. It is based on the LD50 and NOEL. But in the maximum cases, you will know the therapeutic dose, so therapeutic dose criteria will be used. Then 10 ppm criteria is fixed contamination limit. Industry default, worst case check. So 10 ppm criteria is a worst case check and can be used by default, but out of all these three, the most stringent MACO value to be considered, that is lowest MACO value, is required to be considered. Now understand regarding the swab sample acceptance limit and the rinse sample acceptance limit based on the MACO. Swab sample limit is equal to MACO into SSA into SRF divided by TSA. MACO is the lowest value from the or out of three criteria like therapeutic, then toxicological, and the 10 ppm criteria that we have to put here in this formula. SSA is swabbed surface area. Typically it is 25 centimeter square. You can consider. TSA is total equipment surface area. SRF is swab recovery factor. So swab sample acceptance limit can be calculated from this formula. Then rinse sample acceptance limit. Step 1, equipment specific MACO, and step 2, final rinse limit. So here the MACO is used for the equipment and also for the total train. Train means number of equipments which are being used in the manufacturing of that product. For example, if you are working into a granulation, then compression, film coating for a tablet formulation. So all the equipments involved in this manufacturing will make a train or will make a list. So it is the MACO of total train. SAS is surface area of specific equipment, RV is rinse volume in ml, RRF is rinse recovery factor. So based on this, you can calculate the swab sample acceptance limit and the rinse sample acceptance limit. So key points for the MACO calculations are to be considered that always select the lowest MACO from the three criteria so that you can justify the regulatory agencies that you are working on a very stringent MACO. Swab and rinse calculations ensure equipment cleanliness. Follow GMP, FDA and ICH cleaning validation expectations. Use and understand the guideline principles. Recovery factors help in realistic acceptance limit. In conclusion, MACO is a core of residue limit setting in the cleaning validation. It combines scientific rigor and patient safety, ensures no cross-contamination in multi-product facilities. So nowadays, cleaning validation, MACO calculation and justification is becoming very important from the regulatory point of view, from the regulatory audit point of view, from the quality audit point of view. And it provides the information and confidence to the auditors that your cleaning methods are proper, your cleaning validation is up to the mark and as per the requirement of the regulatory standards. So as an industry professional, we should understand regarding the MACO, how it is calculated and what is the quality requirement. Thank you for watching the video.